going to go over cell organelles for biology. This is going to be a long video, so I'm going to put up here the timestamps. So if you want to watch certain parts of the video, you can jump to that part. If you want to watch all of it, you can. If you want to come back and do small chunks, that's great. All right, so organelles are going to be part of a cell. I have two main types. I have prokaryotic, which are very basic. I have a nucleoid. I have a circular DNA membrane in here. I have plasma membrane. I have a cell wall. I have a flagellum on some of them. And I have these free ribosomes that are inside these prokaryotics. These are super basic, very simple organisms. When you look over here at eukaryotic, you can tell they're a lot more complex. I have a lot more going on inside of the cell than I do here with my prokaryotes. So we're going to talk about all the different types of organelles that are in these more complex cells. The first one we're going to talk about is plasma membrane. This here is a plasma membrane. I can see I have these phospholipids. Remember the macromolecule lipids. These are going to be part of that macromolecule. I have a hydrophilic head. These are going to be water-loving. I have a hydrophobic tail. These are going to be nonpolar. These are going to be in the center. This is going to be a watertight area. And they call this a phospholipid bilayer because I have two layers. And you see how they're oriented. This part here will be an aqueous environment outside of the cell. This will be an aqueous environment inside the cell. And this is going to allow for the ability for the cell to be able to separate from its environment. This is also what we call compartmentalization, where it is allowing it to be able to process inside the cell and the organelle certain parts of it and not interfere with other things that are going on outside. This is going to regulate material. So this is going to allow material to pass through. It's going to allow proteins, these are proteins, these are going to allow material to pass through these proteins if they have specific charges, specific types of properties. The next one we're going to talk about is the nucleus. This is a nucleus, and then inside I have a nucleolus. I have a double membrane. This is going to contain nearly all the DNA in the cell. I have this double membrane right here. Remember that plasma membrane we just looked at? That's the same thing I hear. Each of these lines is one of those double membranes. So here I have two. So I have two double membranes. So these are two layers of those phospholipid bilayers that's going to be selective. So it's going to be selective going through the first one, selective going through that second one. They have pores. So these pores are going to allow material in. So you see these little petals here on the side. Those are pores that are going to be part of what we call this nuclear envelope. And so this envelope envelops the chromatin, the DNA material inside the cell. The nucleolus is this part right here. This is where ribosomes are going to be put together, what we call synthesized. So synthesized means to be put together. So ribosomes are going to be synthesized here. Ribosomes are two parts. You have a top and a bottom subunit, and they're going to be used where mRNA can come into this area and it can help to process and synthesize proteins. So this part here gets made or synthesized in this area of the nucleus. DNA also gets transcribed into RNA, so messenger RNA that goes into these ribosomes out. So these little dots right here on this rough ER, these are all ribosomes. And so they get put on. And then mRNA will get created in here. It's going to be sent to these ribosomes. And these ribosomes are going to be embedded in the plasma membrane of these organelles. And then the mRNA is going to come through. It's going to be read. It's going to produce proteins. So this is kind of an area where we want to have a lot of ribosomes. And so the nucleus can get information to it really quick. So let's talk about chromatin. Some people get confused with chromatin and chromosome. Here I have a DNA strand. I have two complementary strands. And you can see my DNA is a helix. I have this curve. The DNA just can't curl in on its own. It would become too tangled up. So you see these little spheres? These are proteins. And these proteins are going to wrap around, this DNA is going to wrap around those proteins. And then all those proteins with that DNA wrapped around are going to coil and coil and coil. And you think of old telephone cords, how they're coiled and curly-cued. This is kind of what it does, and that's to prevent tangling from happening inside. So these chromatin are going to be pretty loose, and they're going to be just kind of in these coils. And this is how the cell is most of the time. Chromatin is just a relaxed form. When it gets ready to reproduce or go into mitosis, then I'm going to start seeing this bunching up of chromatin, and that turns it into what we call a chromosome. Okay. Here I have ribosomes again, 2 subunits. I have a large and a small. mRNA is going to come through, and this is going to help code for DNA codes for proteins. So this mRNA is coding for a sequence of proteins. tRNA is going to come in. I'm going to be able to make this protein sequence because of this ribosome organelle. It's going to be found in both prokaryotic and eukaryotes. This is the only organelle that we have that is what we call non-membrane bound. All the other ones are going to have a phospholipid membrane, and these do not have that. You have 2 types. You have free ribosomes, and these are ones that are just floating around in the cell. So you see these little dots? These are ribosomes. These are floating around inside the cell, and they're just making proteins for the cell's needs. So if something in here needs a specific type of protein, this ribosome will be able to make it. The bound ribosomes, you see this little maze of material right here, these little dots all around it, those are called bound ribosomes, and those bound ribosomes are going to make proteins that can be used and shipped outside of the cell. So these are going to be more specialized proteins. They're going to get packaged by the Golgi. We'll talk about that here in a second. But it's going to leave this, what we call this rough ER, and it's going to go through the Golgi. But this is going to be a special type of protein that's going to leave. Now, not every single one that's made, but a large majority of this will leave the cell. The cell can use some that are made by the rough ER. All right, so my 2 types of endoplasmic reticulum, we also call them ER. I have the ref. The ref is attached to that nucleus, because remember, this makes the ribosomes. The ribosomes will come out here. They're going to embed inside this rough ER. I have mRNA gets made. The mRNA is going to come out here. It's going to hit these ribosomes, and it's going to start making proteins. Protein factory is what it is. Now it's going to make it, and then it's going to be able to ship it outside of the cell or ship it to the Golgi body. So it's going to make proteins that will be released or secreted from the cell. The smooth ER does not have those ribosomes. So here I have the rough ER. See all those little dots on my little ribosomes? This one does not. So the smooth ER has a different function. This one's going to produce lipids. It's going to be involved in detoxification. Drugs and certain types of compounds will get broken down inside of this. So if you take Tylenol or some type of medicine, your body's going to try to break it down into a friendlier thing. Next, I have the Golgi apparatus. So here I have the rough ER, and you can see this rough ER, this little bubble right here. This is called a vesicle. And this vesicle, you see that little stuff inside of it? This is a protein that was made by one of these ribosomes. And this ER is trying to send it to the Golgi, and the Golgi is going to take it and it's going to modify it. It's going to fix it. It's going to add stuff to it. It's going to take things off. It's going to tag it with certain molecules. And then it's going to go through, and then it's going to leave, and it's going to come out. But let's talk about these vesicles for a second. These vesicles, that little circle right there, is plasma membrane. So you remember that plasma membrane we saw, that phospholipid bilayer? That's what this is made of. And you need to think of these kind of like soap bubbles. If you've ever played with soap, like in the sink or in a tub, and how you have big bubbles and you can pinch certain ones off and you can like make smaller bubbles, that's a plasma membrane too, and it works the same way. So this ER can pinch off one of these plasma membranes and turn it into a tiny little, almost like a soap bubble. That soap bubble is going to come and it's going to go into the Golgi body. This is a directional, which means it's always going to go inside a cis phase. And as it goes in, it's going to go through these little stacks. And these little stacks don't really connect. So this vesicle is going to come in. It's going to be absorbed by this plasma membrane. So this is like when you're in a tub and you have a bubble and you put it with another bubble and you kind of like push them together and they turn into one big bubble. This is what that plasma membrane does. It's going to turn into a bigger bubble. This protein comes in, enzymes, certain material are going to get added, taken off. It's going to turn into another vesicle, go into the next pouch, and it's going to keep doing that till it's fixed, till it's the way it needs to be. When it leaves, the Golgi is going to pinch off a membrane, and inside is going to be that fixed or that modified protein that was made earlier. If it needs to be used by the cell is going to use it. If not, it's going to go to the plasma membrane of the cell. You see this part right here? It's going to be absorbed into that plasma membrane, and it's going to open up. And this opening is going to allow that to release. When it releases, this now becomes part of this new plasma membrane. So I can enlarge my cell. I can shrink my cell by adding and removing this plasma membrane. I can bring material in, it can pinch off, go places, or vice versa, it can come out. So it's going to produce proteins that were made by the RefER. They're going to get modified. They're going to be shipped, so they're going to go where they need to go. These are flat sacks. I used to call them like pancakes. They're like pancake stacks. And the next one's going to be lysosomes. So lysosomes can actually be made by the Golgi. So when they pinch off, I can make these little bubbles that are going to become lysosomes. So let's talk about those. So they're going to bud off from the Golgi, and they're going to have a very acidic digestive enzyme. So if something gets digested, it gets broken down. And so what's going to happen is these little pieces are going to hydrolyze. So do you guys remember what the word hydrolyze means? Hydro means water, and lyse means to cut. So if something gets hydrolyzed, water's going in and cutting those bonds. It's breaking it down. So it's going to go in and it's going to do that. It can break down lipids. It can break down proteins, carbs. All of these can be recycled by the cell. So if something comes in and it's not functioning properly anymore, the lysosome will go. It's going to absorb it. It's going to go around it into the membrane and become part of that membrane. It's going to release this hydrolyzing enzymes into the inside of that cell or that organelle, and it's going to start breaking it down. If it can reuse things like carbohydrates or lipids or amino acids, it's going to pull it apart and then take it to those parts of the cells that can use it. If it can't use it, it's going to take it to the plasma membrane and dispose of it. It can break down and remove organelles that are no longer needed. So if a mitochondria starts to not function right, or something's wrong with it, or if it's just wore out, the lysosome can go and start to break it down. These are only found in animal cells. Plant cells don't have these. There are some vacuoles that kind of act like a lysosome, but they're not a true lysosome. Now, this has a thick membrane, and this thick membrane protects it from other parts of the cell from destroying and accidentally hydrolyzing an organelle that's fine. But what's interesting is this has a property that if it accidentally bursts open, remember this is very acidic, the inside of the cell is very neutral. It has a pH of around 7. And this enzyme does not work well in this pH. So this is like a safety mechanism. If this busts, it's not going to destroy the whole cell, which is a good example of what we call compartmentalization. It's going to allow this to function properly inside, but it's not going to allow it to properly function outside of that environment. Peroxisomes. These are going to be ones that are enzymes that are going to remove hydrogen and atoms from various substances. It's going to transfer them to oxygen. So I have these right here. These are peroxisomes. And what they're going to do is they are going to make hydrogen peroxide. Okay. And again, these are not produced by the Golgi. Lysosomes are produced by the Golgi, but peroxisomes are not. So they're going to form hydrogen peroxide. And hydrogen peroxide is H2O2. There's 2 oxygen atoms instead of 1. Water has 2 hydrogens, 1 oxygen. Hydrogen peroxide has 2 hydrogens, 2 oxygens. And that makes it hydrogen peroxide. And inside the cell is extremely toxic, but it is used to break down certain materials. And when it does, it can break itself back down into water and oxygen. So this is used to break up fatty acid tails. It's used to help mitochondria with ATP, and ATP is the energy currency of the cell. So it's able to break down some of these fatty acid tails to help with energy. In the liver, it can detoxify alcohol, other harmful compounds. So again, it's a toxic material. So it's able to go in and break some of these bonds apart. Okay, vacuoles, these are going to be storage. This is going to hold things like water, salts, proteins, and carbs. I'm going to find them in plants. We're going to talk more about the plant vacuole here in a minute. But I can also have them in animal cells. And animal cells are going to be used more like a food type of storage. And these are going to be kind of like vesicles. So you remember those little soap bubbles? This is kind of what a food vacuole is. So they're going to be small, and they're just going to be kind of around different areas inside the cell. And they're going to store and move material that the cell needs. So if it needs some type of mineral, if it needs some type of vitamin, if it needs some type of compound, it's going to be able to ship it wherever it needs to go. There are contractile vacuoles, and these are ones you see in paramecium. These are going to contract, and when they contract, they're going to push out water out of this paramecium, and they can allow for it to move. So it could be a motion. It can also be for water balance. So it produces a homeostasis effect inside of this paramecium. Next one, these are one of our energy cells. This is a mitochondria. Here's an electron microscope of a mitochondria. And you see these little lines? These are cristae. These are where you have a lot of surface area for cellular respiration to happen. So let's look at that. So this is going to be the site of cellular respiration. I have a double membrane. So this white layer here is one membrane. This orange layer on the inside is a second membrane. I have what's called cristae. These are these little curves, these little curls that are inside, these waves. These are cristae, so all of this inside is a cristae. I have matrix, which is the gel inside that cristae. And then I have these ribosomes. These ribosomes are very primitive, very basic, like almost bacterial type of ribosomes. And these are going to be making proteins for the cell. They also have what's called mitochondrial DNA. So see these little circles? These are very similar to bacterial DNA. They're going to be circular, not linear, like regular DNA. And these are also what we call maternal, which means these got passed down. All DNA from mitochondria get passed down from the women in your genetic lineage. So your mom, your aunts, if they're on your mom's side, all of you guys all have the same type of mitochondrial DNA as your mom and the females in her line. There can be 100 to 1,000 in just one cell. So one eukaryotic cell could have 100, it could have 1,000. If it's a very high functioning metabolic cell, it's going to have a lot. If it's not very fast, if it's not very metabolic, then it's not going to have as many. It's still going to have a lot, but it's not going to have over like 1,000 or something. All right, cytoskeleton. This is going to be network of protein filaments. These are going to be used for transport for materials within the cell. It's going to help maintain shape and support. And so you think of it kind of like poles and cables inside of a tent. If I lay this tent down, it's going to be flat. It's going to have no shape. But if I put in these tent poles, if I put in rope, if I pull on it, I'm able to give it a very specific shape. And this specific shape is going to allow it to have a specific function. So here you see these little fibers coming off. These here are parts of that cytoskeleton material. They're going to anchor, they're going to help hold stuff together in place, and they're not going to allow it to move around. So let's talk about the first one. These are microtubules, and these are pretty big, and they're basically like straws. They're going to be big little straws. They're going to have they're going to help you separate chromosomes. So when we talk about centrioles, these are only found in animal cells. These centrioles, you can see these little tubes, these are microtubules. and we have a triplet of microtubules that go around in a circle. And these are going to help pull chromosomes to opposite ends of the cell when we do cell division. So here I can see I have centrioles. I can make these microtubules. There are these spindle fibers. These are going to go, these tubes are going to come down. They're going to collect chromosomes, and they're going to start pulling them back and forth. So these are going to be used in cell division. I can also see them in flagella and cilia. So here I have some flagella. Flagella are little tails that will undulate, or undulate, which means to move, or they're going to have cilia, which are going to, they call them, they beat, and they're going to push and back, and we can use them for motion. We can use it for food capture, or they can sweep it into their mouths. So these are little tubes that are able to work for certain types of functions. The next one we have are microfilaments. These are going to support structure to the cell. And again, think of it kind of like a tent. So this is poles. These are going to go in. It's going to provide that shape and structure. Contraction of muscles is going to allow stuff to pull together, and it's going to allow stuff to pull apart. All right, there is a third one. It's called intermediate filaments, and they are just kind of like a mesh network, like a netting that kind of helps support other materials. So we're only going to talk about two. There's three types, but microfilaments and microtubules are the ones you need to know. Next thing we're going to talk about are plant cells. So plant cells have very specific organelles that are just made for plants. So plants do not have lysosomes. Remember, these are my recycling. They do not have centrioles. These are those microtubules that are used in cellular separation. They're not in flagella. So plants don't have these. Plants have all of these. Now, there are some things in plant cells that animal cells will never have. And that's going to be these. Plant cells will never have a cell wall or chloroplasts or a large vacuole. They have a small vacuole, and it's more like a food vacuole. A large vacuole in a plant works a lot different. You have what are called chromoplasts. These are colors. And then you have amylioplasts that are going to be kind of like starch that's going to be inside the plant. So here I have a cell wall. This is a cell walls under a microscope. And you can see they look like little bricks. They're like little Lego bricks. And they're going to stack on top of each other. And these are going to give support. They're going to protect the inside of the cell. So it's going to prevent water from evaporating out too fast. It's going to keep it straight. Because remember, plants don't have a skeletal system. They have to find a way to be able to support themselves without a skeleton. So these cell walls are able to do that. And they're able to do it because they're made of cellulose. So you remember which macromolecule cellulose is made from? It's carbohydrates. So these are the ones that are kind of a different type of bonds. They're really hard for humans to break down. We don't have the enzymes to eat grass. And so that produces a pretty flat, kind of a very nice, structured, sturdy type of material. They're going to be next to the plasma membrane inside, so the cell wall's on the outside, and you have a plasma membrane on the inside. Because remember, all cells have a plasma membrane. So when you see here, this is my cell wall, but see this little layer here on the inside? That's my plasma membrane. So the cell wall is going to protect, but my plasma membrane is still going to be very selective about what it lets through. The cell wall has these holes called plasmodesmata that are going to allow material to get from one cell to another. So one cell is just not cut off from everything. But this plasma membrane is going to choose what's able to come in and what's able to come out. So let's look at this. Here you can see this gray line. This is like a cell wall. And these holes, those are those plasmodesmata I was talking about. They're going to allow material to pass back and forth. But you see how I kind of have stuff pulled away from that cell wall? This is that plasma membrane. So this plasma membrane sits inside of that. I have a large vacuole, and so when water starts to evaporate out of this vacuole, this plasma membrane is going to pull in. It's kind of like taking like a sandwich bag and filling it up with water and putting it in a box, and you make it real tight inside that box. and then you let some water out, and then the bag kind of gets kind of inside, and it's kind of like that. That's also why when plants don't have a lot of water, they start to wilt. That vacuum inside that plasma membrane is starting to pull away from that cell wall, and it's not giving it support. And we call that turgid pressure. And then when you water it, it fills that vacuole back up, that plasma membrane squeezes back up against that cell wall, and it causes the plant to perk back up. So your vacuole, you have a central vacuole. So this is how much space that central vacuole takes. So when this starts to deplete with water, this is going to pull in. And when it does, that plasma membrane is going to pull in with it. And this is going to cause the plant to not have that support or structure inside that cell wall. These are only found in plants. Turgid pressure or turgor pressure is what's going to cause the plant to perk up. When it doesn't have it, it's going to start to wilt. These are going to store organic molecules, so I can store water, I can store certain types of ions, certain compounds can be inside this vacuole for the plant to use later on. We have also what are called plastids. I'm going to talk about 3 types. These are all going to have double membranes, similar to mitochondria. Mitochondria is double membrane, remember. They all contain their own DNA, which is similar to mitochondria. So we're going to talk about chromoplasts, chloroplasts, and leucoplasts. So chloroplasts, these little green little circles right here, that's chloroplasts. And I can tell because they're green. This is going to be the site of photosynthesis, and this is what's going to happen in autotrophic cells. So an autotroph is an organism that uses sunlight to produce glucose. It's going to make some type of food source. It's going to absorb that energy and convert it into something usable, for example, glucose. It's going to have a double membrane, so each of these little circles has a double membrane. So here's an example of what it looks like. So this is these little green coins look like this. I have that double membrane, outer membrane, inner membrane. I have these little, I call them like little Oreo cookies. These little Oreo cookies are called a thylakoid. And if I have a stack of thylakoids, we call it a granum. And then inside the stack, I have these openings inside of these thylakoids. And I have a thylakoid lumen inside, and it's like a gel-like structure. I have a thylakoid membrane, so when I open that up, I see this membrane, and that's where we're going to be looking when we talking about Krebs cycle. That's kind of what we're looking at, is that the stroma. We'll talk about that too then. This is on the outside. This covers all of these thylakoids, all these granum. It coats all that. Out here, I'm going to have ribosomes. I'm going to have DNA for the replication of this chloroplast later on. So membrane-bound compartments, these are all going to be inside my thylakoids. They're going to contain chlorophyll. Whenever you go and you slide in grass and you get grass stains, those grass stains is you've broken open these cells, and that chlorophyll is getting on your clothes. That's why sometimes it's really hard for grass stains to get out of clothes. You have stroma, that's that gel-like structure, and that's going to be your DNA and your ribosomes for the chloroplast are going to be in that area there. So these are all going to be green. Now chromo means color. Chromoplasts are going to contain any color but green. So you're going to find these in fruits and flowers. So the apple skin, that's going to have chromoplasts. This flower petal are going to have chromoplasts. If I took these and I ripped them, and I was able to take a fine layer of this and looked at it under a microscope, it would look something like this. So you see these little dots, these colors. This is what makes these flowers and these fruits have these colors, or these chloroplasts, which are colored chromoplasts, are going to be in there, and they're going to produce that color. That's the color that you see. Leucoplasts or that one's so hard to say. Amylioplasts. Okay, these are going to be ones that are going to have no color. They're going to be white. So here is my leucoplasts. And then these are going to be starch filled. So a potato, this part here is going to be that plastid. And then this is going to be all type of starch. Okay. All right, that was a long one, but we've got through it. So those are all the organelles you need to know for regular biology class. Talk to you guys later. Bye.